Hello everyone, welcome to today's live broadcast, Germline Cancer Genetics in 2015. We are delighted to bring you this educational web seminar presented by Laboratories. I am Judy and I'll be your moderator for today's event. We have a few important announcements before we begin. This webcast is designed to be interactive and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Judy Garber, MD, MPH. Dr. Garber is the director for the Center for Cancer Genetics and Prevention, Susan F. Smith Center for Women's Cancers, Dana-Farber Cancer Institute. I will now turn it over to Dr. Garber for her presentation.
Good afternoon. Thanks for joining me for this discussion of germline cancer genetics. I know that a lot of the lectures in this series are about really the technical issues in genetics and genomics, and today we're going to talk a little bit about some of the clinical issues that are raised by evaluation of germline genetics. And as this slide says, since I changed the title slightly, many of those things are intentional, of course, and some are more inadvertent. I do have some conflicts of interest to disclose, and those are related to clinical trial work with agents for patients, women with breast cancer and ovarian cancer related to BRCA1 and 2 mutations. It is certainly the genomic era, and although I haven't used a slide like this in many years, when Angelina Jolie announced that she was having prophylactic surgeries because of a hereditary predisposition to cancer, she really did change the face of cancer genetics in many ways. And internationally, there has been an increase in the number of women pursuing genetic testing, and this has been sustained since this announcement now 2 years ago. Recently, her ovaries were removed as well. Most of what is known about hereditary predisposition or genetic risk of cancer comes from the early model work, all mathematical, by Alfred Knudson and Louise Strong. And they identified patterns of cancer and hypothesized that some individuals were born with a predisposition to cancer, not cancer itself, and that this was because they carried a mutation in one copy of paired genes. And they expected these to be genes involved in regulation, not in structure. And time has proven them correct. The first gene cloned that conferred risk of cancer was RB, the gene for hereditary retinoblastoma. And in rapid succession, P53 and a number of other genes came along, and now there are hundreds of hereditary predisposition genes, and often there are genes that perhaps you may not have known about, or syndromes that were not so well recognized. Tumor suppressor genes are those which in their normal function really prevent cancer. When one gene is altered, in fact, function is generally normal, although there are some dose effects. When the second copy is lost in an individual cell, then there is much more propensity for DNA damage and accumulation of error, changes in passage through the cell cycle, and much cancer predisposition presents this way. This is not the only kind of cancer predisposition. There are some genes that are oncogenes inherited in the so-called on position, so you can think of tumor suppressor genes as the brakes and oncogenes as the accelerator, if that's easier. This is an example of a family that might present in which a hereditary predisposition could be active. The patient with breast cancer is female. Females are always circles on pedigrees, males are squares. This is not my convention. If the symbol is colored in, generally that's the disease of interest. The arrows indicate the patients of interest. So the 32-year-old with breast cancer has a family history on the paternal side with multiple individuals with breast cancer, including a male with breast cancer and a woman with very early onset disease. Cancers generally occur when they're solid tumors, at least in individuals late in life. 29 is quite young. This woman also had ovarian cancer. On the mother's side, the mother also had breast cancer, but there are multiple female family members who had no cancer at all into late ages, so one would be more concerned about the paternal lineage. This is the Where's Waldo cartoon that many may recognize. In the book, Waldo was a character who appeared on every page, difficult to identify from everything that looked around him to be other people. Once you knew where Waldo was, however, although Waldo is, whoops, let's see, there he is, then obviously one could look exactly the same place every time, and there he'd be. So in patient testing, one tries to test the individual in a family with the highest chance of carrying a predisposing mutation, and once that mutation is identified, other family members can be found to either share the mutation or not. If a mutation is present and a family member does not share that mutation, then they also do not share the hereditary risk. We suggest that features of families that might indicate that a hereditary predisposition might be present include, of course, multiple cancers in family members. This can be challenging for diseases like breast or colon cancer, which are common in the population, so having multiple affected family members is not rare. You can use age then to help decide. Two family members with breast cancer in their 70s certainly don't confer the same risk as two family members with breast cancer in their 20s and 30s. Multiple primaries suggesting increased susceptibility to cancer, sometimes bilaterality of tumors, multiple renal cell carcinomas, for example. Some tumors are more often bilateral, like ovarian cancer. There are multiple, in these described syndromes, you can see constellations of tumors together, so breast and ovarian cancer in the hereditary breast ovarian cancer syndromes, or colon and uterine cancers in Lynch syndrome, for example. You look for multiple generations. If the families are small, you may not see them, and there may be associated findings, such as congenital anomalies or sometimes specific tumor histologies. Many patients have been tested in the past and were negative, but you might have reason to retest those patients. And these would be, for example, if you always wondered whether there was a mutation because of family history, or that new family members are affected, or the patient herself or himself develops a second or third malignancy. If genetic testing changes, so we've been hearing in your other lectures about next-generation sequencing by and large, I imagine, and other technologies that have been able to look at large numbers of genes more frequently or more easily, and that has certainly changed the way testing has been offered in these settings, and I'll talk about that some more. There are clinical guidelines about who might be tested, and these have changed over time. And then, as I'm sure you've heard, if you're studying tumors, so you're looking now at two genomes, the somatic genome of the tumor that's acquired a number of mutations, and the original genome of the individual, the patient perhaps, who was born with certain mutations. If you study only the tumor, you may not be able to tell whether some of the mutations identified originated in the patient or the tumor, and we'll discuss this some more. So let's talk, for example, about a condition that we know a lot about, because that is changing as testing becomes more widely available and more comprehensive. So we've had 20 years to study the BRCA1 and BRCA2 hereditary breast ovarian cancer predisposition genes. And you can see that already the names are not completely helpful because they sound like breast cancer genes, and they are associated with the highest risks of breast cancer in BRCA1 or 2, but also with ovarian cancer, more in BRCA1, with a small but real risk of male breast cancer, very small but certainly real risk of pancreatic cancer, and increasingly data that there is a form of prostate cancer that is more common in BRCA carriers, and this would be a more aggressive form of prostate cancer. So while there is not necessarily an earlier age at diagnosis, the tumors themselves are more aggressive and are more likely to appear across the age spectrum. Whoops, nothing's happening. I think we're frozen, but I'll try again. So my slide will not advance. Let's see what happens if there's another way to do this. So my slide is not moving. So if I'm going to disappear for 1 second and just see if it's the connection to my mouse. Hold on. So I'm sorry I can't advance the slide. Judy or Don, do you have a way to help with this? Well, I can talk to you through the slides because I can see them, but I don't think you can see them. Why don't we just keep going, and hopefully they'll find a way to fix what's happening here. I'm hoping you can still hear me. So I just wanted to mention that there have been guidelines for the management of patients with these risks of cancer that have come because of all the work that's gone on before. Now recently there's been a paper from Tim Rebick and colleagues using data from the SIMBA consortium. So one of the ways that, oh, my whole screen is frozen. Whoops, there we go. No, thank you. Okay. Thank you, Judy. So I can't move the slides still. Well, I'm not sure what to do, but I guess they'll take it under control. So in any case, there is evidence of genotype-phenotype correlation. This means, of course, that mutations, let's see. Well, hopefully this will be working better now. Yes, look at that. Okay, hopefully we're back on. So genotype-phenotype correlations. Well, there are tens of thousands of individuals with identified mutations in BRCA1 and BRCA2. And Tim Rebick took these data from this large consortium to which most active researchers around the world have contributed and has identified, as you can see, some variation in risk associated with specific location of mutations in BRCA1 and 2. And the question is whether this will be something that has clinical utility, and the expectation is that it will. In addition, there are modifier genes. So by studying individuals with the same mutations who develop cancers at different times of life, one can look and see whether mutations in other genes, such as hormone metabolizing genes or carcinogen metabolizing genes or SNPs at locations of unclear function, might affect breast cancer or ovarian cancer risk, and they do. These two are easily tested for and could be used for clinical purposes. And there's so much data available now that you really can, for example, demonstrate that MRIs add to screening for early cancers and are better than mammograms, even in women as they get older and their breasts should get easier to image. And that prophylactic surgery is effective at reducing the incidence of breast cancer for sure, and that the surgery itself can have good cosmetic results so that women can find the option more acceptable. And the very powerful data that removal of the ovaries and fallopian tubes really almost eliminates the risk of ovarian and related cancers. Now, not completely, because there are still related tissues in the area, but this is helpful since there's no good early detection for ovarian cancer. And you can see that more than reducing the incidence of cancer, using genetic information of this power can help to reduce mortality as well. So people who have their ovaries removed are less likely to die not only from ovarian cancer but also from breast cancer. And data from the group here at the Brigham has identified early precursor cells in the fallopian tubes themselves, suggesting that ovarian cancer does not necessarily develop in the ovaries, but actually in the fallopian tubes next door. So there are a lot of ways to do studies like this. If you're interested in the genomics of all of this, the most important issue is that you have the appropriate specimens to work from. And some of these conditions are quite rare. So for example, in the era of targeting EGFR mutant lung cancers with agents that can exploit this weakness, the specific mutation T790M is a common mechanism by which these lung cancers develop resistance to the EGFR inhibitors. But there is a small group of patients who have a T790M mutation before ever receiving the targeted therapy. About half of those patients can be shown to have a germline or inherent mutation in EGFR at T790M. And those patients appear to have familial lung cancer risk. So here's a pedigree with multiple lung cancers, 3 in one generation, and the mother and the maternal grandfather and his brother, and the mutation can be identified not only in the tumor but also in the germline. And now you can use the internet to collect families where individuals have this mutation because they know that they do. This issue of studying the tumor to get access to the germline is perhaps a little reversed from what we had expected, but certainly germline genetic information is helpful. Now, for many tumor analyses looking for acquired mutations to target with therapy, it is helpful to know the germline so that you can subtract out mutations that have been present inherently in the individual and are not acquired by the tumor and therefore not good therapeutic targets. To geneticists, this is sort of a waste because you're throwing away all that you've learned about the germline from that part of the analysis. Here, for example, is a paper from Ellie Van Allen in the group here as part of the Stand Up to Cancer project where prostate cancers were sequenced looking for targets in hormone-resistant tumors. And you can see that in the group with the standard genomic profile with PTEN mutations, which are known to be common, and PIK3CA mutations, that targeting the mTOR pathway with PI3 kinase and AKT inhibitors can be quite effective. But within this project, they identified a patient with a BRCA2 mutation in his tumor, and when we tested his blood, he did have a germline mutation in BRCA2. So instead of targeting his PTEN system, his therapy targeted the BRCA2 deficiency with investigational drugs like PARP inhibitors and approved drugs like platinum. Most recently, Velkulescu from Hopkins looked at this issue of screening tumors just by looking at the tumor DNA and not sequencing the germline. If you go on and use the various databases to try to assign significance to all of the sequence variants that are found in the genes that you choose to examine. So you usually have to screen through and eliminate genes that are too common because you will be spending years curating the data set. But if you can look at those that are, for example, less than 1% and try to subtract out what you assume to be germline mutations just using databases, you will have a very high rate of misclassification. If here's just using the COSMIC database, you can get a sense for the amount of error that can be made by misunderstanding. Here at Dana-Farber, where we have an existing program called Profile that's used for these for tumor sequencing, this is a study that looks at approximately 300 genes known to be important in cancer and cancer predisposition. Though we don't routinely do the germline yet, we do a lot of curation that accounts for possible germline influence, and this kind of more thoughtful curation can help us to avoid at least some of the problems that one would otherwise encounter. But here, for example, is a patient, this young woman here, who had a thyroid cancer at 21, which was analyzed in the Profile project by the OncoPanel technology, and had a BRAF mutation identified, which is quite commonly acquired in thyroid cancer and was not remarkable, and had an ATM variant that is also quite common in the general population, so not necessarily part of this syndrome, but had a BRCA2 alteration identified in 38% of reads, and not 50%, but still high enough that when she had germline sequencing, she was found to have a BRCA2 mutation. Now, that mutation would have been expected to have existed in a family with a history of breast and ovarian cancers, but as you can see, she has quite a small family and a lot of males on her father's side, not much on her mother's side, and an unaffected mother. There is a breast cancer back here. There's another thyroid cancer that's not usually part of the syndrome. So we have not yet figured out exactly what this means. We certainly would not have tested her based on this information, but now we know from her tumor sequence that going further, she has a germline BRCA mutation. However, most physicians are more comfortable focusing just on the somatic mutations of the tumor and looking at targeted therapies, and are not so interested or comfortable thinking about the germline implications. These are data from Stacy Gray here at Dana-Farber, surveying oncologists here in the Harvard system who nonetheless were much more comfortable with somatic than germline mutations. And there is a lot of work going on for those of you in the research community asking if you look at the germline as well as the tumor, or if you're not in cancer and you're looking at the germline, are you ethically obligated to return all the results that you find? And that really depends on the very specifics of the consent form that your research subjects should sign, because generally the feeling is that you are not obligated to return information about incidental findings, so things you weren't looking for. If this is a study of mutations looking for contributions to ALS, then you don't have to talk about genes that predispose to hypertension. But you have to be very clear in what you agree with or contract with the patient initially before the specimen is collected and the analysis begun, so that you are both clear about what you will and will not disclose. There are whole groups studying issues like this in the American College of Medical Genetics and elsewhere. So I wanted to talk for a few minutes about a project that's ongoing at Dana-Farber as part of the CSER group. This is funded by the National Institutes of the National Human Genome Research Institute. And here at Dana-Farber, this project is actually led by Levi Garraway and Pasiani, but I have been active as well. This is a study. Oh no, we're back. If you could advance the slide. This is a study of patients with, patients with colon and lung cancers. I don't think these slides do this all the time, but you never know. Anyway, colon and lung cancers who are offered the opportunity to have their tumor sequenced. This is sequencing the whole exome and also the exome of their blood for germline material. The goal is for them to have full sequence of tumor and germline material so that the germline can be subtracted out, but also that they have the option of learning results of both tumor and germline, or they can opt out of one or the other. I mean, in general, if you don't want your tumor sequenced, you wouldn't be in this project. Could you try again, please, to advance the slide? Okay, well, so what the next slide shows was that in the first 113 patients, there were numerous mutations, perhaps 5 identified per tumor, and the vast majority of these were returned to the patient's physician for use in trying to identify targeted therapies that could be used in the management of these patients with advanced lung and colon cancer. On the germline side, there were also hundreds of mutations or sequence alterations identified, but only a few % were returned to the patient's physician. And that's because most of the findings were not in genes where they would have clinical significance. But, of course, most of the significance is not for the patient himself, or herself, since their usual problem now is advanced cancer, rather than the risk of other unrelated diseases, but there is implication for family members. And this is why people might consider germline genetic testing. If we did have a patient who actually had an actionable germline mutation in a very important cancer predisposition gene, but that patient had declined in advance to receive the results of his genetic analysis in his germline, and therefore we were unable to disclose that information to him or to his relatives. So I don't know whether you can do anything about the frozen screen. Well, so we'll try to go on here. I have slides that show some of this more graphically so that you can see the kinds of genes that were identified in the screening. But you're just going to have to take my word for it because I can't make any of these slides come up. I don't know if anyone else can. Oh, Judy's going to try again. Let's hope for the best. Okay, let's see. Well, that's something, but now I can't see the rest of the slides. Oh, maybe they're coming back. Let's give it a minute here. I apologize for this. Okay, now they're back. They're back. They're not moving. Well, this is actually a test of me to see if I can actually still give a talk without having the slides in front of me. We'll see if that works. Well, so for those of you who are visual, I apologize. But I'm 233, 34, 37. Can we? Maybe it's just slow. I don't know. Well, so I'm going to talk now about genetic testing that is not available only in the research setting, but also available in the clinical setting as part of commercial testing. And this would be slide 37, Judy, if you can get it back on. So about 3 years ago now, perhaps, Mary Claire King at the University of Washington looked at a group of 360 patients with unselected ovarian cancer, and by that I mean patients with ovarian cancer but not selected for family history in particular, and these patients, thank you, these patients, as you can see, had mutations not only in BRCA1 and BRCA2, but also a few mutations in each of 12 other genes. So a quarter of the cohort altogether was found to have a predisposing mutation potentially. Now the scary part of this from the clinical perspective was that 30% of the women with mutations did not have family history, and almost 40% were older than 60 at diagnosis. And as I told you initially, most hereditary predisposition is present in individuals diagnosed at unusually young ages. So if we could advance the slide, I can tell you that, or next, there you go, that the patients, that the other genes were genes that were part of DNA repair pathways that had been identified once the function of BRCA1 and BRCA2 were clearer, so that these genes, which are similarly important in repair of DNA errors, were looked at, and many of them found to be associated with breast cancer in families when mutations were present in the germline. This came along at the same time. Next slide, please. As the ruling by the Supreme Court that a single company could not own the patent to genes and could not own the genes or have a monopoly on germline testing. Next slide, please. And that led to the development by many companies now entering the space of technology for testing multiple genes in the same assay. So this is not really meant to be read, but this is an analysis or a list of 5 products, or 6, I guess, where next generation sequencing is used to analyze the germline for a larger and larger and ever enlarging group of genes. So until this time, testing was limited to BRCA1 and BRCA2 in the breast cancer arena, to about 5 genes for Lynch syndrome in the colon cancer families, and a few other genes. Not much commercial testing took place for some of the rarer syndromes. But with the move to next-generation sequencing and the end of the testing monopoly, perhaps, it became possible for the same price to test for an ever-growing number of genes. Next slide. These genes tend to be grouped, let's say, so the numbers are totally arbitrary, but the breast cancer genes include genes you may be familiar with if you're at all interested in cancer biology, genes like P53 or more recently PALB2, mismatch repair genes for colon cancer, polyposis genes now with the addition of the POL genes and several others. So we've gone from panels of 14 genes to panels now up to almost 50 genes, with expectations for getting to 97 genes in a panel offered in the UK. This is very efficient testing, not that you're looking at all of the other genes in which you're not interested, so forgive the double negative, but these are cancer-related genes, but you can look at a large number of them for a much more reasonable cost. Next slide, please. The challenge of this has become partly definition. So when we have previously discussed syndromes, we're generally talking about clinical syndromes where a group of cancers might be seen in the same individual or perhaps the same family, maybe at young ages, the features that I gave you before. Now we may find an individual with a family history that's not nearly as compelling, but in whom a germline mutation is found nonetheless. And the question is whether the cancer risks are the same for two individuals with the same mutation, one with and one without a family history. Or does the definition of a syndrome now require or depend entirely on the presence of a mutation, or is clinical information still useful? Next slide, please. So we did a small study with Nadine Tung at Beth Israel showing that the addition of what we're calling panel testing, these expanded panels of genes rather than individual candidate genes, has led to an increased frequency of detection of mutations. Next slide, please. But one example of the conundrum that I just described is here a very classic family with what is called the Li Fraumeni Syndrome. And here you can see the individual with breast cancer at a young age with a sibling with multiple primary tumors. These are rare tumor types: adrenocortical carcinoma, osteosarcoma, soft tissue sarcoma. By 26, having had 3 cancers, this is rare. Here's a lung cancer, but at 48. So this is a classic family, and most people would recognize this as Li Fraumeni Syndrome, genetic test or not. Next slide, please. But in this family, there is an individual, next slide, tested at age 48 after being diagnosed with pancreatic cancer. In this individual's family, there are other gastrointestinal cancers, another pancreatic cancer, another GI cancer, and a brain tumor, all of which could be the Lynch syndrome. And this individual was tested in the panel as an efficient way to look at the multiple genes that predispose to Lynch syndrome, and no mutation was found, but a mutation was identified in P53. The rest of the family, so the father was tested, he also was negative for Lynch syndrome and negative for P53. So this either is coming from the maternal lineage and we know nothing about her side of the family, or this is a new mutation in this patient. But we generally would not have tested him previously for a P53 mutation based on this diagnosis. So we're missing some of these, and the definition of Li Fraumeni syndrome now, is it that terrible history I showed you on the previous slide, or is having a P53 mutation enough? And this comes up now because healthy individuals are being tested on these panels, and unexpected mutations are being identified. Next slide, please. This matters for things like P53, where there are data that individuals with the mutation and a very high lifetime risk of of cancer overall, multiple cancers, as you can see, may benefit from having screening with a whole body MRI. This is not a technique that's available everywhere. It's still largely a research technique. It does not include radiation. It does not include contrast, so that makes it easier to do. And we haven't yet proved that it worked, but at least we have something to offer individuals, that is research, to at least find their cancers early, though we can't prevent them at this time. Next slide, please. One problem, as you would anticipate, in looking at the germline of individuals, if you're doing whole exome or even large numbers of known genes, is the frequency of unspecified variants or alterations whose pathogenicity can really not be definitively determined using all of the available databases and even functional data at this time. And the more you look, the more you find. So this is just a frequency count from 198 patients at Stanford who had their analysis done, and there were certainly more variants than there were individual mutations. This is fine, but you really can't tell people what they mean, and of course they will be present in family members because they're sharing DNA. Next slide, please. These are just our data from a recent count. This is the last 6 months of genetic testing being conducted at Dana-Farber. You can see that we have moved from focus testing to panel testing very frequently, and although we have certainly found mutations in about 10% of individuals we've tested, we have at least twice as many variants of uncertain significance to go with those findings, so people are not always very happy for having spent the money on the test. Next slide, please. Oh, insurance does cover this, by the way. There is a new project called PROMPT. This is another example of trying to use the internet to accelerate research. So this kind of testing is available clinically and commercially all over the country. That means that primary care physicians and gynecologists can order genetic testing on their patients. It's only a blood test after all. But when these uninterpretable results appear, we're asking patients to consider registering in this online database to provide their information and perhaps accelerate the pace of discovery and risk assessment associated with these genes. So we want to establish penetrance, for example, and we'll need large numbers of individuals with mutations that are indeed quite rare in the population. Next slide, please. So there are risks and benefits, or benefits and limitations of genetic testing, as we've talked about. They're efficient, they're less expensive, you can look at multiple genes at the same time. Sometimes, however, you find things that don't seem to explain the family history in front of you, and you're not always sure what to do with that information. Next slide. And you can have this particular problem where you do this kind of next generation approach, and what you find in an unsuspecting individual are 3 germline mutations. This may be the record. One in BRCA2 that perhaps could link to this maternal aunt with a great aunt with ovarian cancer. One in PMS2, which is a colon cancer or Lynch syndrome gene that might account for this endometrial cancer, although we can't see much else to go with it up there. And MUTYH, which is a common gene in polyposis. So this person certainly did not expect all of this, and we now have to go back through the family, find out how prevalent or what the distribution of these mutations is, since we don't know whether they all came from one side or some from each side or both sides, and that this information then can be shared with the relevant family members. Next slide. So I wanted to shift gears here and talk about another application of genetic information in cancer patients. This is not really so much based on the kind of genomics that we've talked about, but just some of the recent data that genetic information can determine the behavior of tumors that develop in mutation carriers and therefore can affect treatment decisions. This has led to a lot of pressure for rapid turnaround of genetic tests. So in the past, if you would recognize that someone might be a carrier at diagnosis, they could have their adjuvant chemotherapy and all of their other management before their final surgery, and the results would come back in 6 or 8 weeks, and that would be enough. But now you really want those results back in 2 weeks or faster. So this is a small discussion about breast cancers in individuals with BRCA1 or 2 mutations, and of course, for most of them, at diagnosis, the valuable potential use of the information might be in choosing to have mastectomies instead of more conservative surgery, this to avoid the risk of additional primary cancers, not so much as a difference in managing the new cancer. And this issue of when to take out the ovaries, since some tumors are hormone-driven, removing the ovaries, especially in premenopausal women, can affect not only the risk of new ovarian cancer, but can also be therapeutic for managing the breast cancer itself. Not much change for radiation or HER2 targeting tumors. Next slide, please. Well, there you go. So it is surprising, I think, still, that if you do molecular analysis, these are of breast cancer subtypes looking at arrays, gene arrays, that you can see that for BRCA1-related tumors, the majority, 70%, do not express either estrogen or progesterone receptor or HER2, and most breast cancers are characterized for all of these. But BRCA2-related tumors, 70% of the time, express both estrogen and progesterone receptor, but not HER2. And the P53 tumors, these are the Li Fraumeni tumors that I presented to you before, they are generally either estrogen receptor positive or HER2 positive 2/3 of the time. And it's puzzling, I suppose, in part as well, because the BRCA1-related tumors almost always have a P53 mutation in the tumor, although the germline mutation is BRCA1. So here these tumors have a P53 mutation, but they are a completely different subtype. And whether these arise from different precursor cells or there's something else about them that changes their differentiation along the way is completely unknown at this time, at least as far as I know. Next slide, please. So we said before that BRCA1 and 2 are genes that are important in DNA repair, and they have a specific role in the repair of homologous recombination in that pathway, in the repair of double-strand breaks in the DNA, although they have roles in multiple functions in the 5 DNA repair pathways. And here's some data from Dan Silver and David Livingston here at Dana-Farber, showing that in a BRCA1 deficient cell that's been radiated, as you might expect if there's double-strand break in the DNA, that when the chromosomes reanneal that you can have some unusual forms. And here is the same unusual form found in a tumor from an individual with completely normal BRCA1 in their germline, but in the tumor the same evidence of DNA damage is present. So perhaps a different mutation is present in this tumor, at least, maybe in the germline as well. Next slide. So our group was interested in whether there were therapeutic implications for this, and we had the following model that individuals with a BRCA1 or 2 mutation would have breast cancers that were of the triple negative type and with defects of DNA repair, but that there might be a subset of other breast cancers that were intact for BRCA1, but somewhere down the pathway had similar DNA repair deficiencies and might benefit from the same strategies, exploiting those weaknesses. Next slide. There was also cell line data. This is the first BRCA1 - tumor cell line compared to tumor cells with intact BRCA genes, and they showed much better killing with the drug cisplatin, which is a DNA cross-linking agent thought to cause double-strand breaks in the DNA, as compared to doxorubicin, which is these are drugs that are used much more often in the treatment of breast cancer, to which the BRCA tumor cells were resistant, but the other tumor cells more sensitive. Next slide, please. And we had some data from ovarian cancer treatment that women with mutations in BRCA1 and 2 who got ovarian cancer actually did a little better than non-carriers. It's not a lot better, but it is better statistically significantly. And the question was it because their tumors were more sensitive to the effect of platinum, which is a standard agent in the treatment of ovarian cancer? Next slide. So we designed a small trial. This is a neoadjuvant trial. For those of you in research, the advantage of neoadjuvant studies is the patient is diagnosed, they receive their systemic therapy before surgery, so the tumor is still in place. If the tumor shrinks or disappears completely, then the chemotherapy was likely the right stuff and was effective, and then the patient goes to surgery. You can collect tumor before surgery, and you can collect persistent tumor at surgery, so you can have a lot of specimens to work with. And we did a small trial of 28 patients with triple-negative breast cancer unselected for their BRCA status. We found that 10%, well, 6 had their tumors disappear completely, 10 had very good clinical responses. Those younger women did, and both of the women in the cohort who turned out to have mutations had their disease disappear as well. And this is what you would have expected if our hypothesis was correct, that these were the tumors that would respond to platinum. Next slide, please. Stephen Narod used the same protocol of platinum to treat women with BRCA1 mutations. These were treated in Poland, where there is a common founder mutation in the population, so an easy and inexpensive single-site genetic test on these women at diagnosis, and 60% or 61% of those women had their tumor disappear completely with 4 doses of single-agent cisplatin. Next slide, please. So this has led to some increased activity in the therapeutic world, looking at the value of cisplatin, which is actually carboplatin, but these are old platinum drugs from long ago, now asking, do they have a role in breast cancer treatment that we might have missed the first time around? And these are data showing that the addition of carboplatin, and this is the German version, increased the response rate from 38 to 58%, which is quite a statistically significant improvement, and the same in the American version of this trial, where the comparison was against chemotherapy without platinum and without the addition of a drug targeting angiogenesis. Next slide. And the recent German data showed that in the 315 patients, or 295 of them who had triple-negative breast cancer, so-called, and could be looked at for their BRCA status, that much of the benefit, much greater, an 83% response rate in women with carboplatin compared to those with who did not receive carboplatin, and not nearly the same difference, although a difference in women with family history, and much less difference in those without a germline mutation at all. Next slide, please. And a new trial presented in December by Dr. Tutt from the UK, in which women with metastatic triple-negative breast cancer were given either carboplatin or a taxane, the other active drug, and they did crossover at progression if they wanted to. Next slide. And this study showed, you can advance again, that really most of the benefit of platinum was in individuals with a germline mutation, not in those without, and that in the group that did not have a germline mutation, platinum might have been perhaps a little less active, although not statistically significantly so, than the taxane. But the big advantage here to those with a mutation at diagnosis. Next slide. But there are old data to suggest that these tumors that are based, after all, on DNA repair deficiency might be sensitive to other forms of chemotherapy, and it may not be the platinum at all. So these are old data from Israel, where there are also founder mutations, where in this case, fixed tissue, paraffin-embedded tumor tissue from a cohort now about 25 years old were looked at with single-site testing for the 3 founder mutations, and the group divided into those with and without mutations. And those with mutations did just as well as those without, as long as the BRCA1 patients had chemotherapy. This was old-fashioned chemotherapy, not platinum, not anthracyclines, not taxanes, and they did just as well. Next slide. And data from the MD Anderson group showing that those with a mutation did better in their studies of individuals treated with taxanes and anthracyclines, the standard drugs, but not with platinum. So they did well in that case in addition. Next slide, please. I promise we're almost done. So we have a small trial here and at multiple sites around the country looking at comparing patients with mutations and triple-negative or high-grade ER-positive disease at diagnosis, randomizing them to platinum or the standard of care, and we'll see whether these tumors really are more sensitive or just more sensitive to chemotherapy in general. Next slide, please. Next slide, please. So the PARP inhibitors have been developed, I guess you'll have to hit it again, to look at exploitation of part of the DNA repair machinery, not just the double-strand breaks that platinum does. PAR is an enzyme important in DNA repair, and the inhibitors bind PAR to the DNA and make it difficult for the machinery to progress and repair the errors. They convert basically single-site defects into double-strand breaks. These are data just showing that one of these, olaparib, was effective in individuals with mutations at the time they were first treated for metastatic disease. Treated for metastatic disease even though they had received a number of other agents. So you should get the sense from this CAT scan that there's a mass here and that mass is smaller in the second screen, and the same here where there's a large mass that didn't belong, and that is absent here, and these are just the response rates. Next slide, please. And other data here, again, this in a trial for individuals who had BRCA mutation, so the first time that having a germline mutation determined eligibility for treatment. These individuals had lung metastases. This one had lung metastases that really almost disappeared, and these are the number of people responding to therapy. Next slide, please. And the data here showing activity in ovarian cancer at 2 different doses. Next slide, please. These are mutation carriers. And here, showing that while in ovarian cancer, these drugs work both in tumors from people with germline mutations and without germline mutations, so some more common shared mechanism. In breast cancer, it's really only people with mutations who respond to these agents. Next slide. We're almost done. So there are at least 5 drugs in this class that are in development at this time. Next slide. One of the obvious trial designs was for single-agent PARP inhibitor to be compared against any of a number of standard drugs that can be given as the first-line treatment, so at first metastatic disease. So unfortunately now there are 3 competing trials, each with a similar PARP inhibitor in a 2-to-1 randomization against the drug of choice, and we hope that at least one of these will actually complete enrollment, so we will be able to answer this very important question. Next slide, please. And there is the first of these, oh, I'm sorry, you can skip this. Well, you can leave this slide because it really shows that these drugs are being tried not only in breast and ovarian cancer, but also in pancreatic cancer. There are trials in prostate cancer, the tumors that can occur in individuals with a germline mutation. So it suggests that in some of these individuals with family history at least, or perhaps with specific histologies, that we will be testing for germline mutation to try to expand the therapeutic window for these people with tumors that often respond to little. Next slide, please. This is an adjuvant trial, so drugs that work in metastatic disease are often moved earlier in treatment. So now this is the first PARP inhibitor study for women who have triple-negative breast cancer, a germline mutation in BRCA1 or 2, who have completed all of their other therapy, either chemotherapy before surgery or after surgery, radiation, whatever, and then are randomized to 1 year of either the PARP inhibitor or a placebo, seeing whether one can delay recurrence or prevent recurrence entirely with the use of these drugs in this setting. Next slide. This is a large international effort because it will take 1,300 women to do this study, and this trial is up and running. Next slide, please. Oh, we can skip these. Next slide. Just that you can combine these drugs with other chemotherapeutic agents and increase the response rates. Next slide, please. I'm sorry, too much chemo, I think. An interesting mechanism of resistance for those of you in the targeted therapy arena where resistance is the problem. And here in BRCA carriers, it is possible for a mutation to be acquired in the existing germline mutant copy that restores the protein almost completely and therefore restores function of the gene BRCA1 or 2, whichever. And this mechanism then, these tumor cells that have reacquired a germline, tumor cells have reacquired a functioning BRCA1 or 2 protein can then become resistant to platinums and PARP inhibitors. This has been shown in ovarian cancer and breast cancer largely by the Seattle group, and accounts for about 20 or 30% of resistance to these agents. Next slide. But there are data that some of these tumors remain sensitive to platinums even after PARP inhibitors, and the mechanism of this is not clear. But there are many mechanisms of resistance, so we'll see. Next slide. These are just other combinations that are in trials. Next slide. I promise we'll get back to genetics in just a minute. You can skip this. Next slide. And CDK1 inhibitors. Next slide. And you can even make tumor cells like lung cancers that were never sensitive to PARP inhibitors possibly sensitive at this time by inducing errors with the CDK1 inhibitor. Next slide. And there are efforts, of course, to predict sensitivity to these agents, platinum and PARP inhibitor, by looking for various defects, looking at the tumor, not at the germline, since some of the tumors with acquired mutations in BRCA1 or 2, for example, can be sensitive to killing by these agents. This is from our little study showing that the tumors with either germline or acquired tumor mutations or methylation of BRCA1 would be sensitive to the BRCA, I'm sorry, sensitive to the to the platinum drugs. Next slide. And this is work from Andrea Richardson looking at using the predicted telomeric changes that one would expect if you have double-strand breaks in the DNA. When the chromosomes re-anneal after these errors, they might not be in the proper orientation. And if you just count up the amount of telomeric error, that can be a score of how well these cells do and don't repair their DNA errors. This was called non-allelic telomeric imbalance, NTAI, and this is part of a tissue-based assay that's now being studied to see whether it predicts response. Next slide. In our data presented at the AACR meeting recently, it did predict for platinum response, but there are other data sets in which this, as part of the HRD assay being studied by Myriad Genetics, did not predict responders. This was true in the TNT trial that I talked about earlier. Next slide. I promise we're almost at the end. Right, so back to genetics. So genetic testing has risks and benefits, and it's important for people generally to have thought about those before they have genetic testing. There is direct-to-consumer genetic testing available at this time. There's a company that does SNP analysis, but that includes founder mutations in BRCA1 and 2, and that information can be made available to people without having to go through their physicians. There's a new panel testing company doing next-generation sequencing originally at this point for 17 genes, but I'm sure with plans to expand. These include BRCA1 and 2 and genes for other strong predisposition syndromes like Lynch syndrome. And for $249, you don't need a doctor. You can call them up and order your test, and they'll have a physician do it for you. This is color genomics for those of you interested in checking them out. Next slide. We talked about this patient already. Next slide. You do have to be prepared for the fact you may find things you wish you hadn't. And then there are a lot of people who worked on some of the projects I presented today, and these are the genomic projects with the help of the Broad Institute. Next slide. And the triple negative projects with the help of this group of individuals at Mass General, Dana Farber, Beth Israel, and in Spain. Next slide. And for most patients, in the end, it comes down to this, to trying to understand the risks and benefits of genetic information, the possibility of erroneous information, which definitely happens, and how this information can be used for their health. Thank you for paying attention and for making it through all of those little technical errors. And I hope you learned something this afternoon that was of interest. Okay, there are questions. Oh goodness, okay. So I'm reading these. Are the P53 mutations in the BRCA1 cases loss of heterozygosity or point mutations? So the P53 mutations that are seen in the tumors of BRCA1 carriers are acquired mutations, and they run the gamut, as far as I'm aware, from point mutations to large deletions. P53 as a gene has a number of types of mutations. The difference between Li Fraumeni mutations are not always the same type of mutations as somatic mutations. Well, you know, there can be germline mutations that run the gamut as well, from point mutations to rearrangements and deletions. I guess one is more confident of deletion mutations as being functionally significant. It is a problem in P53 that many of the mutations are missense mutations in the germline as they are in tumors, and therefore sometimes figuring out the functional significance or using bioinformatics to figure out the significance leaves you with the inability to determine for sure whether some of these missense alterations are clinically significant or not. People use the tumor data absolutely as much as the germline to try to assign significance. It's the advantage of cancer, I guess, compared to diseases that have only one germline to work with. So there's no straight answer to this, and there are the usual efforts with large databases and consortia to try and assign significance to mutations both in the germline and in the somatic space. Is there a difference in the response to PARP inhibitors if tumors have certain P53 mutations? In part, the question since individuals... this is a question about P53 and telomerase. So as far as I am aware, PARP inhibitors have not been shown to be particularly effective in individuals with Li Fraumeni or germline P53-related tumors, although I am actually not aware to what extent they've been looked at. One would not expect them to be particularly effective in that group, but I really don't know that for sure. Telomerase is a very interesting feature of many of these and other tumors, and I'm not sure that its role in predicting response to P53 has been looked at. Its role in looking at cancer susceptibility to various things has certainly been looked at, and very intriguing data, and its role in interactions with P53 probably among the most intriguing data, but I don't think any of those things have yet been linked to PARP inhibition. That doesn't mean they won't be. Maybe you'll do it. How are telomeric errors being measured? So telomeric errors are really not part of this kind of germline discussion. There are many groups looking at telomerase and telomeric errors, scoring these using genomic technologies. Probably the group furthest along is at the National Cancer Institute, Steve Chanik and Sharon Savage, but I'm afraid that I am not sufficiently conversant with their approaches to tell you. Naturally, Elizabeth, the Nobel Prize winner who identified telomerase, also has done a huge amount of work in this area. I can't believe her last name has left me for the moment, but she's at UCSF. I'm sure you can find her. And I can't remember the answer to your question, but you should. Oh, that's so embarrassing. I can't remember her last name. It will come to me, I hope. Well, I don't know how long you want to wait for other questions, but maybe I've exhausted you all, and if so, I appreciate your paying attention here to the bitter end, and I hope you find the presentation useful. Judy? Oh, Elizabeth Blackburn, thank you so much, Don. She is the Nobel Prize winner who won for her work on telomerase.
Thank you, Dr. Garber. Today's webcast will be available for on-demand viewing through November 14th, 2015. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Bye.